Can you hear me okay? In the back? Good? Okay. So a cancer-free world. Think about that as you hear the rain. Think about who you know who has faced cancer, and think about what a cancer-free world could mean to you. I think about a cancer-free world all of the time. I think about how to get to that cancer-free world. And what I want to talk to you about today is how Ohio State is using science to take us to a cancer-free world. And to do that, we have to go back in time a little bit to understand where we were and where we're heading. Sir William Osler, often considered the father of internal medicine, once said, if it were not for the great variability among individuals, medicine might as well be a science and not an art. So what does that mean? It means that we're all different. With different cancers, we're different. And if we can understand the basis for that disease, we can treat it better. We can do a better job beyond the art. To go back a little further, in terms of how science can help us classify biology and human disease, we look back to the 1600s when Anton von Leeuwenhoek used an early microscope, and he pointed it at small microorganisms and used that microscope to classify them. And in a similar way, we've used a microscope that you see here to classify cancer. So cancer from the lung is different from cancer from the breast and cancer from the brain. And we use that to help decide how to take care of patients. And so this microscopic cellular classification of cancer would continue for the next 300 years, and it does today too. And what changed in 1994 for me is not only did we have a microscope, but I started to learn about genetics. As a student here on this oval, I studied genetics from 1994 to 1998. I remember hanging out in the oval, and what I learned about genetics then was that it was helping us define cancer differently. I learned that genetic changes, called mutations, could give cancer two of its main properties. One, that cancer grows in an uncontrolled fashion when it's not supposed to. Two, cancer travels throughout the body and invades organs where it doesn't belong. And so these genetic changes could drive that process. What I also learned about genetics as a student was that genetic research was taking place to uncover these driving events in cancer. And it took a team of 10 people studying a handful of genes in one cancer to do that in 1994. I graduated from Ohio State and started medical school in 1998 here. And I saw a different classification for cancer than I was expecting. What I saw was this picture of lung cancer, which was a cellular description of lung cancer. So in one part of the pie chart is adenocarcinoma, square to round cells under a microscope. Another part is the squamous cells, so flat cells under a microscope. And then finally, large cells. And this was the classification that we were using to take care of patients with lung cancer. But at the same time, I knew that genetics research was happening and helping us redefine it, but it wasn't coming to patients yet. It wasn't in practice. It wasn't changing medicine as it should. I graduated from medical school and a PhD here at Ohio State. I started my medical training, oncology training, and in 2008, some miraculous things had happened in that time span. Genetics research had changed because the technology we use for DNA sequencing, or the reading of DNA, had gotten 1 million times faster and 1 million times cheaper. So if you put that together, that's 1 trillion times better. And that technology was changing our view from a cellular view to a molecular view of cancer. So this is a picture showing you a couple of these classifications, and I'm going to walk you through it. On the left is structural rearrangements where two genes can come together, form a new gene called a chimera, or a gene fusion. Chimera is the Greek monster. And this gene fusion can drive cancer growth. And another example, a copy number alteration. So instead of 2 copies of a gene, a cancer cell could have 100 copies of a gene, and that could drive cancer growth. So HER2 is an example you may have heard of in breast cancer. The third example is even a single letter change, 1 letter out of 6 billion, can change a gene in a way to drive cancer growth. And another example is not just the DNA, but the DNA has to be transcribed into a message called RNA. And RNA can help us understand and classify cancer differently. And so this molecular classification was shifting our view of cancer. What was happening, though, the research was happening in a laboratory away from patients, and it hadn't come to patient care yet. And so these technologies were available, and I was using them in 2011. And one of my mentors, a pathologist who actually doesn't see patients, said to me, "Why aren't you using this for your patients, Sameek? Why don't we do it?" And I could only come up with 10 reasons why we shouldn't or couldn't. It's too complex. How are we going to pull off all of this sequencing and interpret it and analyze it? How am I going to use it? If I don't understand it, how do I explain it to my patients? And if we find something, how are we going to find new treatments for them? So all of these reasons, and we decided to do it anyway. In May of 2011, we enrolled our first 2 patients, and by November, we published this paper showing how to do it to bring genomic sequencing to patients with cancer for the first time. The way we were able to do it was through collaboration. We were able to work with radiologists, genetic counselors, pathologists, oncologists, genomic scientists, and computer scientists to collaborate towards a common goal. The next step, though, was not only using this research tool to classify cancer differently, but we needed novel ways to test patients. We needed clinical grade diagnostic tests. And so to make this transition, we needed to innovate. And I want to walk you through my view of innovation through an example about moving people. So you can imagine trying to move somebody in a taxi, and you can think that's very feasible and has some impact, not super high impact. On the other hand, you can think about a teleporter with Scotty and Mr. Spock. And transporting somebody has tremendous impact, but you all giggled and you thought, that's not possible. And we've thought that before. We thought that in the 1960s about travel to the moon, putting a person on the moon. That was felt to be impossible. And the way that happened was, again, through multidisciplinary collaboration. And Ohio State has helped us accomplish the same for novel diagnostics in cancer. So our team here today, you'll meet them in the back later, is developing novel diagnostics, collaborating with specialists in oncology, genomics, computer science, radiology, genetic counseling, cancer biology, drug development. Everyone is working towards this common goal together. And this is our new picture of lung cancer. So the picture you saw was the one I learned about in 1998. But this is the picture we talk about today when we see a patient with lung cancer. So we not only classify by the cell type, which is still important, but the genetic subset of their cancer. And you can see in the green, 20% may have 1 gene mutation, but you can also see these smaller pieces of this pie, much smaller than 1%. So it tells you how important individualized identification of these genetic lesions are for a patient. The other part of this pie chart that's important is the blue unknown. This means we have more work to do. We have more discovery and more characterization of these genetic subsets. When we find these genetic subsets with our novel diagnostics, we also have to find new ways to treat patients. And so we've shifted in clinical trials of novel therapies from a one-size-fits-all, one type of cancer getting the same therapy, to a genetic classification, what we call genomics-based clinical trials, and we match a patient to a therapy based on a genetic marker. It turns out, if you do this, you're more likely to respond to the therapy, spend less time taking ineffective therapies, avoiding toxicities, and people live longer. And so this is what innovation has been able to do at Ohio State, heading towards a cancer-free world with novel diagnostics, genomics-driven trials. Ohio is not a stranger to innovation. The Wright Brothers had 3 principles for innovation. First, new ways to look at old problems. Second, taking calculated risks. And third, working together. And so this is our research team developing diagnostics. We're lucky to be in an environment with the James Cancer Hospital, with the Wexner Medical Center, collaborating in this environment to innovate and bring a cancer-free world to reality. And so I want to thank you, my Buckeye community, for working with our research team and family, because you're part of that team. So we're working together for a cancer-free world. Thank you.
Sameek, thank you for that. It was a wonderful talk. I want to, first of all, note you're 40 years old. So a couple of observations. One is Star Trek was filmed before you were born. So we appreciate your
I watched it in a rerun with my brother.
Yeah, I understand. Gene Roddenberry would be very pleased today. But second on that same note, the Human Genome Project, which sequenced the human genes, actually only finished in 2003. So a first question is, you see the velocity, the tempo, the speed of applying genomics at a cancer center like the one at Ohio State, the tempo. Do we see cures on the horizon soon, or the clinical trials, that this whole area of precision medicine? Can you expound on that?
So 2003, the Genome Project ends, new technologies come about, they're available for research in 2008, and in 2012, we're offering them to patients now. And so we're now able to classify patients differently, match them to novel therapies, and again, when we can do that, therapies are more likely to work, we're more likely to have an improvement in survival, and ultimately that's our goal towards a cure. And each individual cancer, though, may be a different mission and a different project, and we've got to solve that.
And actually, Dr. Drake was asking me, first of all, when is the rain going to end? But the other thing he asked was, if you could predict that. But the other one is, do we have data in terms of living longer, in terms of the application of these targeting techniques? I wonder if maybe, are you part of the Orion project, or are you familiar enough with it? Maybe you would explain.
So 2 things. One, to answer your question about, is genetics improving survival? So a group analyzed about 3 years worth of clinical trials, 32,000 patients, over 500 clinical trials were looked at. Some trials use markers, but the majority of trials have not been using markers. So if we use markers, overall survival is better, response rates are better, and progression-free survival is better. So the data is there, and that's only from 2012 to 2014 data. And so we're still living in the new era of a genomic classification. I like to think of genomics as the new microscope, and we have a better understanding of cancer, and so we've yet to fulfill and see the fruits of what genomics can do. The second part is Orion. Orion stands for Oncology Research Information Exchange Network. And so you may have heard Vice President Biden talking about the need for data sharing. And so there's so much information that we have to learn, and when we're talking about 1% of a certain cancer, that means we need to collaborate across the entire country to identify that small percentage so we can develop new therapies for them, because we can't do that in one clinical trial with one person. So the Orion Network is the James's mission to expand, share data, develop new ways to treat those patients, and that we're looking forward to participating in. So.
Ohio State's taken the lead, creating this massive database where size matters because of the heterogeneity of gene mutations. So this is a remarkable breakthrough right here at Ohio State's Cancer Center. Sameek, thanks so much.